Hello everyone. Today we're going to start talking about the Archaea domain, but first let's explain a little about this system of taxonomic classification. It all started with Linnaeus in 1735 when he was the first one to actually make the division between plants and animals and come up with a way to classify living beings using those two kingdoms. Then after that, they added the microscopic one, the protista. Right now, we don't longer no longer talk like that. Uh, I mean, at least we don't include like too much living beings into that classification, into that kingdom. Then in 1925, a big breakthrough come, and it was that we were able to actually make the difference between membrane-bound organelles and the ones that don't have membrane. We got prokaryote and eukaryote. Um, 1938 with Copeland, we got the first division between plants and the protists. Remember, protists are the ones that got the membrane-bound nucleus, but they don't actually are a plant. So yeah, we got that division there. And after that, Whittaker was the first one to divide the plants and the fungi to actually come up with the idea that the fungus is a different living being, a very different living beings. Then after that, 1990, thanks to Bruce, we got this last division, at least the last one we're going to talk here, and it's between bacteria and archaeas. Yeah, if you look under the microscope, they are pretty much the same. But if you analyze the biochemicals that they have, the molecules that they produce and how they obtain energy, well, you will see that that archaeas and bacteria are just as different as you are from plants. So pretty much different. To expand into this, any living being can be classified using um, this linear classification system. So first we can start by talking about domains. The current ones are this one, bacteria, archaea, or eukarya. Depending on the living being that you're talking about, it could be any of these. Then after that, let's just talk about the first column here. We got the kingdom, Animalia, and that means any living being that it's multicellular, that reproduces sexually, and that must eat something else. Yeah, it's not able to produce their own food. They have to move to it. Then we have the phylum Chordata, and that include any animal that got a spinal cord or something remotely similar to that. Well, in this case, fish, reptiles, amphibians, mammals, birds, all of them are chordata. Then we have the next one that is class, mammalia. Any animal that got these special glands for feeding the offspring. Um, and then when we have the order primata, that as you may already imagine, it includes all the apes and primates that we know of. Then the family Hominidae, we got several cousins in that family. If you look over the internet, you're going to find that, for example, bonobos and chimpanzees, that if you ask me, are basically the same species, but well, we're maybe later we can talk about that. Um, and gorillas also are for the hominidae. Um, what else that we can talk about? Well, the homos, the genus Homo, it's a pretty particular type of uh, animal. In this case, us, the human beings. And the last one, the last member of this Homo genus was the Neanderthal, or more or less the other one was the Neanderthal. Sadly, it went extinct. Maybe you carry one of the genes, but uh, if you're a European or European descendant. Um, but right now is the last evidence that we got that that little guy uh, was for the Homo genus. And then, well, species, Homo sapiens, that's us. Um, and well, that's one way to classify living beings using the domain, kingdom, phylum, class, order, family, and genus, species. So right now we're going to start talking about the first domain, Archaea, just to start with some of them. Pretty basic living beings, not that common since they are not like living like from us. They are not pathogens, so maybe you haven't heard about that. They are the linked branch between bacteria and eukaryotes, so they have some complex biochemical function happening inside them. They are unicellular organism. That must be. They lack membrane-bound nucleus, as any other prokaryote. And the cell wall is composed of either linked lipids, called pseudopeptidoglycan. When we talk about bacteria, we're going to mention about that cell wall also. And well, it's found in any kind of habitats that you can think of. Other any kind. Here, well, we are going to divide it into, you remember here, we're talking about domains. Let's talk about kingdoms. For this, we are going to divide it into 3 kingdoms. The Archaeota, also known to be the living beings to live in the extreme temperature and acidity at the bottom of the ocean or in the extremely salty, acids environment they can live on and thrive on. And the next kingdom is going to be Eukaryota. So those are the ones that live in methane and salty environment. And then, well, we got the Archaeota, found only in high temperature hydrothermal environments. Uh, we're going to see example of those, so you don't, well, get lost a little with this. For this specific, we got the Archaeota. These are known as the thermophiles. They can grow and reproduce at temperature of 100 degrees Celsius, pretty high temperature. They're found in acidic soils, hot spring, and near volcano openings. One example of that is the Methanophyros candleri. The other one is the mesophiles, another subclassification of this one. They live in moderate temperature, ranging from 20 to 40 degrees Celsius. They can be found in cheese, yogurt, any other dairy product. The most common one is the Listeria monocytogenes. Then the last one is going to be the psychrophiles. These live in extremely cold temperatures ranging from -20 to more or less 10 degrees Celsius. Because of this, they are going to be found in the Arctic or alpine soil, deep in the ocean water, high in latitude, also glaciers or snow fields, well, any cold temperature that you can think of. One example of this is Cryosebacterium greenlandensis. Remember, we are talking about Archaeotes. Then, well, we have the Euryarchaeota. From this, we are going to talk about the methanogens. They release methane as waste, so they have an important role in the carbon cycle. And they can be used in the sewage treatment plant. Yeah, to train the dark water, the black waters from your home, they can be used for that. One example of that is the Methanocaldococcus janashi or the Methanobri Bacterias midi. Pretty good names. Then we have the halophiles. They flourish in extremely saline locales found in the Great Salt Lake, pretty specific, the Owen Lake and the Dead Sea, the Mediterranean, well, near that. One example of them is the Dunaliella salina, the Halobacterium salinarum. Um, well, next, talking about these prokaryotes, we got the bacteria, pretty simple one. And it's another term we use for the bacteria. The structure, they don't have nucleus uh, in which the DNA is stored. They are enclosed by a cell wall. In this case, it's the peptidoglycan. It's not the pseudopeptidoglycan like the Archaeas. They have an additional layer outside the cell wall that is called a capsule. That's the reason why you need to, when we are taking some antibiotics, you need to combine that with other substance. Um, Most of the case is the clavulanic acid, amoxicillin with clavulanic acid. The clavulanic acid is going to destroy the capsule of the bacteria, and then the amoxicillin is going to kill the bacteria. So you need both of them to actually get some good damage. For the reproduction, they reproduce asexually or sexually, so they can use conjugation, transformation, or transduction. Binary fission is one of those also. It's that asexual reproduction, and they have exponential growth. They can make spores to, well, so the offspring will be born in a different location. They can be either, they can have either the need for oxygen or not. So if they need oxygen, that's for the metabolism, to make energy, they are going to be considered aerobes, and if they don't, they will be anaerobes. For the nutrition, many of them are heterotrophs. That means that they have to eat something else in order to live. But some of them are able to actually get energy from some chemical reactions, so they will be chemolytotroph or maybe phototroph, like the plants. For the shapes, and this is a way to actually name of them. We got either if they got a spherical shape, that's going to be the cocci. If they look like a rock, that's going to be the bacilli. And well, if they look something else, they will be others. For cocci, well, we got different ways for them to grow, to arrange them. We got the diplococci. So if you look under the microscope, they always be in pairs. The streptococcus, that means that they will make chains. If there are 4 of them, there is the tetrad. Sarcina, if there are 8 of them. And if they look like grapes, if they all grow together, that's going to be the Staphylococcus. The most common one is the Staphylococcus aureus. Maybe you already got that disease because of that. From the bacilli, the rod shape, we can have the chain of bacilli, or maybe with some flagels, or they can make spores. Those are the 3 classifications. And then we got the vibrios with one only flagell, all the spirilia that they have these like weird shape, like this one, and several flagels in one direction, and the spirochetes, also very common. From the next one, well, we are going to talk about for the next video, we're going to talk about the eukaryotes, the eukaryotic cells, the classification. So thank you all for paying attention. See you.